Today I'm doing my presentation on cloning. So I'm going to start off with a question. Have any of you guys ever thought about having, let's say, an identical twin and asking them to go to class for you, maybe to do a speech for you or go to work for you, anything like that? No? None of us?
Yeah.
All right, yeah, a couple of us. All right, there we go. All right, thank you. All right, so I bring this up because cloning is something that could be very possible in the near future, and it's important that people know that this is something that could happen. As a member of the community that pays attention to technological advances in the scientific area, I believe that it's important that this is actually something that's known about. People need to know this because if it's something that's actually that could happen, it's something that, who knows, could be possible for everyone. So first off, I'm going to go ahead and talk about the history behind cloning, and then I'll go on to discuss the downsides of it, and finally I'll discuss the benefits of it. So first off, cloning. What is it? So there's two types of cloning. There's therapeutic cloning and reproductive cloning. So therapeutic cloning obviously has a really long definition that's pretty complicated. So I'm mainly going to be talking about reproductive cloning, which is kind of the cloning of humans, or having the production of a genetic duplicate of an existing organism. So basically having a clone. So according to Genetic Science Learning Center at the University of Utah Health and Sciences, in an article from 2014, they list a timeline of everything that's happened basically in the history of cloning. So one of the first entries is from 1885, and it talks about Hans Driesch, who is a scientist that was studying sea urchins. And in the top, as you can see, what Hans Driesch did was, there's a laser pointer on here, right, somewhere?
So that.
Oh, cool. All right, so he had one embryonic cell, which is this, and he realized that by shaking it, that it created two cells, which later developed into two fully functional sea urchins. And so this was kind of like what began cloning and the history behind it. So without even knowing it, he kind of became like the father of cloning. And then in 1996, this is another timestamp on the timeline, Ian Wilmot and Keith Campbell created Dolly using somatic cell nuclear transfer. This is Dolly right here. They actually cloned a live sheep, and it's actually very impressive how they did it. So they did it by using somatic cell nuclear transfer, right there, which I'll explain later. So in the article from 2015, in the Proceedings of the National Academy of Sciences, Francisco J. Ayala stated that somatic cell nuclear transfer, which, as I told you earlier, is reproductive cloning, that's basically how they did it. They created the exact replica of the sheep, and it's kind of like what was one of the most, it was one of the biggest advancements that had actually happened in cloning in a very long time. But now before that, they had done small things like frogs and salamanders, and then this was like a big step towards like something that could become possible, like cloning humans. So now that we know a little bit of history behind it, I'll go on to discuss the downside. So, of course, with the downside, there are many risks that can come from cloning. So a couple of risks are that there's an immense failure rate. The failure rate, this pie graph, is actually from their website. This is the failure rate that occurs. There's also problems with later development and some abnormal gene expression patterns and telomere differences. So I'll go ahead and discuss the failure rate. So failure rate. The success rate is actually 0.1 to 3%, which is an immense, that's a huge failure rate. So 97% of the time, which is a huge amount. So something else is abnormal gene expression patterns. The Huffington Post, they actually found these two girls. These two girls are identical twins. Their DNA is completely the same. But because of these abnormal gene expression patterns, this is obviously a large exaggeration, but notice how they are extremely different, but their DNA sequences are exactly the same. But that's a very large exaggeration, but it could be something as small as just like hair color. So if I had a clone, maybe his hair color would be black, or maybe his hair would be a little bit curly. It could also be very small things. So Jason T. Eberl, a professor at Indiana University, wrote an article on Project Muse from 2010 that kind of discussed some of the risks that come from this. And he basically there's also many other drawbacks, kind of like it could be offensive to certain religions, or it could cause a new need for definitions such as reproduction, because if we can create basically clones of humans, I mean, it can cause a new need for the definition of reproduction. So now that you know some of the downsides, I'll go on to discuss the benefits. So some of the benefits are stem cell research, medical purposes, revival of extinct species, the reproduction of a deceased pet, cloning livestock, drug production, and human clones. Alright, so let's start with Jurassic Park. How many of you, when I said revival of extinct species, thought of Jurassic Park? So that would be an exaggeration. Like, yeah, they could, but they'd have to have like the DNA sequence of a dinosaur to bring that back. But you never know, that could eventually happen. But I mean, that'd be pretty scary. I mean, I wouldn't go. I mean, it'd be cool, but I'm not into that. So something else would be the revival of the reproduction of a deceased pet. So they had, I can't remember who it was, but I read online from the Huffington Post, I believe, that a woman won a prize of having her deceased pet, basically cloned and revived. So this was her cat. Its name is Rainbow, and this cat is the clone of it, but they named it CC, the two letters C and C, and it's because carbon copy, so they nicknamed it CC. But notice how the two cats, they aren't exactly the same, like different eyes, different like hair palettes, sort of, because not every clone is going to look exactly the same. It's not always going to be that they're identical because, again, of the abnormal gene expression patterns. So I'll talk about the drug production as well, and I don't mean like producing like massive drugs and stuff. I mean that, like, let's say, it's easiest to explain with an example. So let's say there is a terrible virus going around and everyone's getting sick and people are dying in the country. So let's say we discover a vaccination, but we don't have it in a form that we can like inject it, but we have it in some sort of edible form. If we take that and put that in an enucleated cell, and we can take it from, let's say, a cow's udder and combine them and then put it back into the cow, when that cow starts producing milk, the vaccination is actually inside of the milk. So when we drink it, we become vaccinized. It's pretty crazy. So now that I'm approaching the end of my speech, hopefully you have a greater knowledge of cloning and the benefits and downsides of it. So again, cloning can be something that happens in the very near future, and it's important that we have some knowledge about it. First, I discussed the history behind cloning, and then I discussed the downsides of it, and third and finally, I discussed the benefits of it. So you never know when cloning could become beneficial to us and how it could help us in the future. Thank you.